lot of times I start the show by saying we're going to be breaking down a huge topic or an important topic or a big topic. But today we're going to be swimming in a really small topic, and that is particles. I wanted to break down USP 788 and 790. These two chapters in the USP cover the testing, acceptance criteria, sampling for subvisible particles and visible particles. If it's your first time listening to the show, hello and welcome. My name is Subhi Saadeh. My goal is mastering medical device and pharma quality by way of combination products. And this show is how I answer questions I'm curious about or talk to experts about topics that I think are really important. So welcome to the show. Before we jump into the rules for testing, the acceptance criteria, the sampling, how the tests are supposed to be set up, and the different methods that are used, I did want to talk about what USP is. It's not as cut and dry as an ISO standard or something like that. USP, or the United States Pharmacopoeia, is a nonprofit organization whose responsibility it is to develop quality standards for drugs. They also work on biologics and other healthcare products, and interestingly enough, are doing a lot more work in the combination product space, particularly if you look at some of the elastomer-related ones that have come out more recently. But there are also efforts like ICH-Q4 because there are different pharmacopoeias worldwide. There's the US Pharmacopoeia, USP, but there is also the Japanese Pharmacopoeia, European Pharmacopoeia, and others, and there are efforts to harmonize between all of them. I think maybe one of the important differences to talk about ISO versus USP is around scope. USP typically focuses on pharma, whereas ISO has broader. You can have an ISO for automotive or an ISO for bridges or an ISO for finance or whatever. At the same time, I think the similarities both are focused on standardizing requirements, but I think where maybe the USP or pharmacopoeias more broadly end up being a little bit more in the details is for things like generic products. You have USP monographs that speak to the requirements for a generic drug product where that specific drug needs to meet because it's no longer on patent and really anyone can make it. And so if that is the case, you need to demonstrate the agreed upon acceptance criteria for a generic drug. And the USP goes into a little bit more detail and has a few different prongs as far as pharma goes. Diving into the USP chapters themselves, USP 788 specifies testing and acceptance criteria for subvisible particles in injectable drug products. These are the particles that are too small to see by the naked eye. These kinds of particles can present issues, particularly if they're in excess of certain particle counts. It's worth exploring why this standard is so important for pharma and combination products. USP 788 covers subvisible particulate matter. In other words, the particles that you can't see with your naked eye. But why is this such a big deal? The drug is injected directly into your bloodstream, so any particle present, no matter how small, can pose a serious threat to the patient. The actual threat is obviously going to depend on the drug product and whether it's IV or intramuscular, what the target is. But the health risks that are typically associated with particles are things like embolisms, immune responses, inflammation, or even more life-threatening complications. So that's why there are stringent standards as far as particle goes to make sure the products are as clean and free of particles as possible. All right, so USP 788 specifies the test requirements, the sampling requirements, the acceptance criteria for subvisible particles. In other words, the particles that you can't see with your naked eye. The presence of particles can lead to significant health risks because we're talking about injectable drug products, so whether it's injected directly into your bloodstream via IV or intramuscular or subcutaneous, regardless of the target, you are introducing whatever's in the container into your body, and it's not going through the enteral tract through your stomach, and all of that has ways to keep you safe. It's not going through your basic immune response, and therefore it can cause complications. Most common, obviously, these things have to be assessed in a product risk assessment, and so it's product based and target based. But at the same time, there are common risks associated with particles, things like allergic responses, immune responses, embolisms, but they can cause serious health risks. And the presence of particles can be life threatening, depending on what they are. That's why these USPs exist with pretty stringent requirements. USP 788 has two methods, method 1 and method 2. The first one, and I think the one that's most common, is the light obscuration particle count test. This method involves passing the liquid, whatever's in the container, through a light sensor, and as particles move through, they block or scatter the light, and the equipment counts the sizes of these particles. It's fast and it's ideal for clear solutions, but may not work for products that are cloudy or viscous or opaque. The second test method is the microscopic particle count test. This test is used when method 1, light obscuration, isn't suitable. It involves filtering the sample through a membrane. And manually counting the particles under a microscope. It's labor intensive, requires operator training and a very clear and requires more guardrails in terms of who can do the test and how the test is executed. The acceptance criteria for USP 788 depends on whether or not you are working with a small volume container or a large volume container. Small volume containers are considered those that are under 100 ml, and large volume containers are considered those over 100 ml. Most syringes are under 100 ml, so if it's in a syringe, it's typically in that place, and large volume parenterals are things like IV bottles, IV bags. So for small parenterals, the maximum number of particles that are greater than 10 micrometers per container are 6,000 particles, and the maximum number of the larger subvisible particles, which are greater than or equal to 25 micrometers, it is 600 particles. And the acceptance criteria allowed is particle size based. For large volume parenterals, it's per ml. The acceptance criteria is set up a little differently. The maximum is 25 particles that are greater than or equal to 10 micrometers per ml, and the maximum for the larger subvisible particles, the ones that are greater than or equal to 25 micrometers, it's 3 particles per ml. And so these limits are critical for ensuring that the injectable products don't pose a risk to patient safety. You'll notice how when we talked about the acceptance criteria, that the number of samples depends on the product type. For example, the USP typically requires a statistically significant number to be tested based on a sampling plan. USP 790 focuses on visible particles, and these are the particles that can be seen with the naked eye. When it comes to visible particles, there's a lot more focus on in-process controls and in-process checks. USP 790 requires 100% visual inspection, meaning every single unit, whether it's a vial or ampule, syringe, IV bag, if it's a product, it's 100% visual inspection, and it must be performed. The inspections are typically performed against a black and white background. Under controlled lighting, there are lighting requirements to maximize the visibility of the particles. Manual inspection is often performed as well by trained operators who carefully look at the container, rotate it along the table to make sure that they're looking for particles that are floating or adhered to the inside of the container. In addition to the 100% inspection, there's also an AQL sampling plan that's typically done at release. Even though batches are expected to be 100% inspected, there's also an AQL sample that's taken and used as release criteria per the USP. And this ensures that any unit containing visible particles are rejected, maintaining a high product quality standard. I'll do another episode on why that makes sense from a quality point of view and of the pros and cons of 100% inspection. But obviously, because it's in the USP, 100% inspection is not always 100% accurate. So just because all of them are being looked at doesn't mean that there isn't an issue there. It just means that you had a trained operator look at 100% of the containers. 100% inspection, just to close this, and I'll do another episode on this. 100% inspection is not a silver bullet. It can be good to do that kind of inspection, but it doesn't mean you're going to catch everything, which is why there's an AQL that's done at release. So a couple more details around the tools and methods. Light tables are tables that provide consistent lighting, reduce glare, and make it easier to spot particles. And operators inspect each container manually and look at it from multiple angles. There are some automated visual inspection systems that are used for particles, and they offer high speed and consistency, but a lot of times there'll be some kind of human verification. At some point, the AQLs are most commonly done by a human operator. So even if you may have 100% visual inspection in line, you would still have the AQL done by a human operator. And then lastly, a couple of maybe more practical considerations for this USP. Operator training is really important. The inspectors have to go through rigorous training because, especially if you've seen a high-speed IV bag line or something like that, the containers are moving quick. Rigorous training is needed for the operators, and it's a skill that involves periodic retesting and retraining to make sure that everybody who's doing that activity is appropriately qualified. And then maintaining clean handling practices and ensuring that not manipulating or causing false positives or contamination by the fact that you're handling the product through inspection is obviously something to bear in mind. So that's a wrap on today's episode. We've seen how these little tiny particles can cause a huge impact and why we have two, I think, really good USPs in place to address them. If you enjoyed this episode, please subscribe on letscombinate.com or on the YouTube channel at Let's Combinate or on Spotify, Apple, or wherever you listen. And if you have feedback for me, let me know.